Hello, my dear friends. I'm Dr. Indrajeet, and currently I'm working as an assistant professor in Department of Microbiology in KTHM College, Nasik. And today I'm going to talk about one of the fundamental and critical process which is a part of central dogma, that is transcription. So in this, I'll be specifically elaborating on the process of bacterial transcription. So for the next few minutes, I have outlined my presentation in the following points. So firstly, I'll give you a comparative overview between transcription and replication. And then also, I will give you a general overview of the gene expression in prokaryotes and eukaryotes. Then I would like to elaborate on the process of transcription initiation and also the features of bacterial promoters. Then we will dig more into the details of the biochemistry of this process by studying the structure and the function of RNA polymerase. Then I would also like to elaborate on the process of elongation. And finally, I would like to conclude this talk by talking about termination and the techniques which are used to study the transcription start site, that is DNA footprinting. So let's start with the comparative overview between the transcription and the replication. So as we all know that the transcription and replication are of vital importance for cell survival. So there lies some similarities between these two processes. The first similarity is the fundamental chemical mechanism. So in transcription, the RNA polymerase is involved. And this RNA polymerase has an aspartate at the active site. So this aspartate amino acid is involved in coordinating with the magnesium ions. And these magnesium ions are in turn carry out the nucleophilic attack of this alpha phosphate onto the 3' hydroxyl group. So followed by this attack, there is formation of this phosphodiester bond and there is a release of the pyrophosphate. So in case of transcription and replication, the formation of the phosphodiester bond and the release of the pyrophosphate are very much similar. The second similarity is the use of a template. So both in the case of transcription and replication, DNA is used as a template. So the third similarity is with respect to the polarity. And polarity is nothing but the direction of the synthesis. So the direction of synthesis in case of transcription is 5' to 3'. And similarly, in case of replication, in both the contexts, that is in the case of leading strand and also in the case of lagging strand, the synthesis happen in the 5' to 3' direction. However, there are some subtle differences between transcription and replication. So the first difference is the involvement of the segment. So in case of transcription, only one segment of the DNA molecule is involved. That is, the one strand of the DNA molecule is only used for the copying into the RNA molecule. However, in case of replication, both the DNA strands are involved in the duplication. The second difference lies in the use of the template. In case of transcription, only one strand of the DNA molecule is used as a template. However, in case of replication, both the strands of the DNA molecule are important for carrying out the replication. So before we go into details of the transcription, I would like to give you the brief overview of the terminologies used in case of transcriptional DNA molecule. So the first is DNA template strand. So the DNA template strand is the strand which is used as a template in order to generate RNA molecule from it. So another DNA strand, which is DNA non-template, is also termed as a coding strand. This coding strand is almost similar to the newly generated RNA molecule, except in the case of uracil residues. So the third difference between the transcription and the replication is the use of primer. So in case of replication, the RNA primer is used to carry out the replication. And however, in case of transcription, there is no involvement of the RNA molecule to initiate the process of transcription. So this basic difference is because of the difference in the biochemistry of the enzymes which are involved. In case of replication, there is a DNA polymerase which is involved, and in case of transcription, it's RNA polymerase which is involved. And the last difference between the transcription and the replication is its accuracy. However, the replication is very crucial for the cell survival. It is a very high fidelity process. In case of transcription, there is one mistake which happens after every 10,000 nucleotides which are synthesized. However, in case of high fidelity replication, there is only one mistake out of 10 million nucleotides which are synthesized. So let's dig more into details of the gene expression. So RNA metabolism in case of eukaryotes is very complex. As we all know that the bacteria lacks the compartmentalization of organelles, the transcription and the translation process are coupled. So as soon as the RNA molecule is formed, the ribosome machinery sits onto the RNA molecule and initiate the process of translation. And after translation, there is a formation of the chain of amino acid, which ultimately folds into the functional protein molecules. In case of eukaryotes, the initial transcription event is very much similar to that of bacteria. However, there is an extensive involvement of the post-transcriptional processing. So as soon as the RNA comes out of the RNA polymerase, immediately it gets capped at the 5' end and the poly A tails are added at the 3' end. So this results in the increase in the stability of this RNA molecule. So this stable molecule is in turn transported through the nuclear pore into the cytoplasm. So as soon as it arrives in the cytoplasm, ribosome machinery sits onto the RNA molecule and initiate the process of translation. And the similarity at the end of transcription in both the prokaryotes as well as in eukaryotes, it's its degradation. So both the RNA molecule in case of bacteria and in case of eukaryotes are degraded at the end. So let's talk more about transcription initiation. So RNA polymerase is the main enzyme responsible for the transcription. But alone RNA polymerase cannot do its job. So this core enzyme, that is the RNA polymerase, can bind to any of the DNA molecule. But this binding is a very low affinity binding, and that cannot initiate the process of transcription. So the RNA polymerase always needs the help of another accessory factor called a sigma factor. This sigma factor in turn binds to the RNA molecule in a very stable manner. So once the binding is over, then this results in the stable complex between the sigma factor and the RNA polymerase. And this RNA polymerase and sigma factor complex goes and binds specifically onto the site of DNA, that is the promoter. And this results in the very high affinity binding, and this results in turn in the initiation of the transcription process. If you closely look at the structure of this sigma complex and the RNA polymerase, this sigma complex has an elongated structure that actually extends along the surface of the core subunit. And when the extension happens, this results in the formation of much stable complex. And that will lead in turn the transcription initiation. So this is the choreography of the transcription initiation process. So as we all know, promoter is the site of the DNA where the RNA polymerase binds. So the initial binding of the RNA polymerase to the site of the promoter result in a very low affinity, that is the cos complex. And after binding of a sigma factor, that results in more stable complex, and that results in ultimately formation of an open complex. This open complex is nothing but the melting of the nucleotides beneath the RNA polymerase. So there is a partial unwinding of the nucleotide beneath the RNA polymerase complex. And that will in turn result in the initiation process. So this initiation requires an interaction of sigma factor with the promoter element as well. The sigma factor has different regions ranging from region 1 to region 4. Out of this, the region 2 and region 4 are very critical for interacting or formation of a stable complex with the -10 and the -35 element of the promoter. And there is additional domain associated with the RNA polymerase, that is the C-terminal domain. And this C-terminal domain is very critical to interact with the upstream element. So all these interactions result in a formation of much stable complex, and that will in turn leads to the transcription initiation process. So let's dig more into the details of the bacterial promoters. So this is consensus sequences for sigma 70. So for this, almost 300 promoter elements have been sequenced and aligned with each other, and there has been consensus which was found with respect to the nucleotide at each position. So on the y-axis, you have nucleotide frequency, which is given in percentage, and on the x-axis, you have position onto the DNA. As we can see here, Tata 80 are the predominant sequences which are present at the -10 region of a sigma 70 promoters. Followed by that, there are 15 to 19 nucleotides of spacer sequence which are non-specific, but the length is quite specific. And upstream of this is -35 element, which has a consensus sequence of TTGA and CA. So these are typical features of wide range of bacterial promoters. As we have just seen, the promoter has a -10 and -35 region separated by 17 to 19 nucleotide spacer region. And in case of rRNA, there are some additional element, that is upstream elements. These upstream elements are important for interaction of C-terminal domain of RNA polymerase with it. And that will lead to, in turn, robust transcription from these rRNA genes. These are typical promoter in case of E. coli galactose metabolizing genes. So this E. coli galactose metabolizing genes lacks -35 sequence and also upstream element, but it has an extended -10 region which just lies upstream of the -10 sequences. There are some genes which uses these promoters which have a discriminator region which lies upstream of the -10 sequence, but just before the transcription start site. So if you closely look at the interaction of RNA polymerase onto this -10 and -35, these are some sequences which are very important for it. So the green colored arrow here depicts that if you modify these sequences, this will in turn prevent binding of RNA polymerase to this -10 as well as -35 region. And the purple arrow here depicts that if you mutate or if you change the sequence of this DNA, then it will completely abolish or sometimes reduce the rate of transcription from it. And black arrow here depicts that these are the sequences which are in turn protected by RNA polymerase against any sort of modifications such as alkylation or methylation, which are important for modulation of gene expression. So what we can conclude out of this is most points of contact for RNA polymerase lies only onto one face of the double-stranded DNA molecule, and the one face is nothing but it's a non-template strand, which is nothing but a coding strand. So let's dig more into details of interaction of the sigma factor with respect to this promoter element. As we have just seen that the sigma factor has a region ranging from region 1 to region 4. Out of this, region 2 is very important to have an interaction of the -10 of the promoter element. And region 4, which is very important to interact with -35 of the promoter sequence. And then this in turn drives the melting of the DNA molecule beneath the RNA polymerase and sigma complex. And these are the amino acid from region 2.4 from sigma factor, which are very, very critical to interact with the DNA molecule. So these are the arginine, threonine, glutamine, tryptophan, and the tyrosine. So these traits of amino acid drives a critical interaction of this sigma factor onto this minus chain region of the promoter sequence. So initially, when sigma complex binds, it almost covers up to 75 base pair. And this initiation of transcription can result in subsequently the abortive process. So in this case, there is initial transcription, but after few rounds of nucleotides which are added, the sigma factor is immediately released, and that results in an abortive process. But once the successful initiation happens, the sigma factor forms around 15 to 20 bases and covers up to 30 to 40 bases. So this is a process of abortive initiation, which mostly happen in case of bacterial transcription initiation process. So let's look into the details of an enzyme which is important to catalyze the transcription, that is RNA polymerase. So RNA polymerase is a huge protein, and it has different regions across it where it catalyzes the entry of the downstream DNA molecule, and that is only carried out through the beta pincer molecule. It also has a non-template channel, so the non-template DNA strand is subsequently channeled through this pore. There is also the template channel where the template DNA strand is in turn channeled through this pore. And there is upstream DNA molecule which is immediately wound as soon as it comes out of the RNA polymerase. In addition to it, there is also an RNA exit channel through which the newly formed RNA exit through this pore. So this is a crystal structure of the prokaryotic RNA polymerase and is compared with the eukaryotic RNA polymerase. As we can see here, prokaryotic RNA polymerase is comparatively much simpler than the eukaryotic RNA polymerase. And both the RNA polymerase structure has alpha helices and the beta sheets connected by the beta loops here. So let's dig more into the process of elongation. So this is a scrunching model which is widely accepted for the process of elongation. So in this model, initially the RNA polymerase sits onto the promoter and it just pulls the DNA through its active site. And this pulling of the DNA results in its unwinding, and the process of transcription elongation can happen. So if you see here, these are the pincers where the DNA molecule enters into the core of RNA polymerase complex. And only 8 to 9 nucleotides are base paired to the DNA template here. As soon as the RNA is formed, the used template is kind of catenated just at the exit channel of the RNA polymerase. So this is a scrunching model. As you can see here, the DNA is pulled into and the new NTPs are added. But as we have just seen in the first slide, there can be some mistakes with respect to RNA polymerase can happen. So there are two proofreading mechanisms which operates in case of RNA polymerase, in case of the wrong nucleotides when added. So when the wrong nucleotide is added, first the nucleotide is removed and the pyrophosphate is added, and then the pyrophosphate is in turn removed and the right nucleotide is added in place of pyrophosphate. And this is called as a pyrophosphorylation editing. Another type is hydrolyting editing. In this case, when the wrong nucleotide is added, RNA polymerase just backtracks and just remove the wrong or hydrolyze the wrong nucleotide. And then we'll look into the process of termination. So this schematic represents the process of termination, and which is row independent termination. And this is called as an intrinsic termination. And this intrinsic termination is solely determined by the sequence which is present on the RNA molecule. So as soon as RNA polymerase approaches towards end site, the RNA sequences in the newly formed RNA result in the formation of this hairpin. And this hairpin has a very typical features. In the stem, it's rich with the GC-rich sequences, where at the base, it has 7 U residues. And the formation of the hairpin in turn results an obstruction for the ongoing RNA polymerase, and this in turn drives the termination. In E. coli, more than the half of the sequences undergoes intrinsic termination. As after sequencing, there has been 1100 sequences of this intrinsic terminator has been found in case of E. coli. Another termination is extrinsic termination, which is normally catalyzed by Rho proteins. In the case of extrinsic termination, the Rho is very important, and the Rho has N-terminal RNA binding domain and the C-terminal ATPase domain. As you can see here, the ATPase domain is normally important for hydrolyzing ATP, and which in turn provides energy for the Rho factor to traverse across the RNA molecule. However, the active form of this Rho structure is an hexameric protein. So 6 molecules of Rho come together and almost forms a donut-like structure. And within this donut, as this arrow depicts, the RNA is pulled along. So let's see the choreography of the extrinsic termination process. So initially, as the RNA polymerase transcription happens, Rho attaches specific sites on the RNA, which is RUT site. So the RUT site name came from Rho utilization sites. And with the help of its ATPase domain, Rho translocate along the RNA molecule and then approaches to the RNA polymerase. Since RNA polymerase here binds or pause at the hairpin, and finally, Rho with its ATPase domain completely unwind the RNA-DNA hybrid, and ultimately it releases all the transcription machinery, including RNA polymerase. And the newly generated RNA immediately get translated by the use of ribosome machinery. And finally, I would like to conclude this talk by talking about the important technique which are used to study the transcription, that is DNA footprinting. As the name suggests, normally it is used to find out the footprint of a protein onto the DNA. So in case of transcription, as RNA polymerase is attached onto the DNA, it is cross-linked by some irreversible agents like formaldehyde. So in case of this experiment, the 5' end or sometimes the 3' end is labeled, and the DNase1 enzyme is used to digest the DNA. So DNase1 is a non-specific nuclease which can cleave every possible residue except the RNA polymerase. And you can see here, these are the fragments which are generated after digestion. And these fragments are in turn run onto the sequencing gel, which has polyacrylamide and urea onto it. So this is a control where no protein is involved. You can see here, there is a label, and the nearest label is towards the end, and the farthest label is on the top. And you can see here the ladder which is formed because of absence of any protein. But in case of test, where the protein is bound, there is resistance for the cleavage, or there is resistance for the formation of these bands, as you can see here as a footprint here. So this is nothing but the site onto the DNA where RNA polymerase is bound. So this is an experiment where TF3A, which is an important transcription factor to initiate the eukaryotic transcription, the footprint for this protein has been found. So in the first lane, you can see here is the ladder, and the hollow it depicts here the site onto the DNA where TF3A is bound. So with this, I would like to conclude this by summarizing what we have looked into until now. We have seen first the comparative overview between the process of transcription in bacteria and in eukaryotes. As there is lack of compartmentalization, transcription and the translation in case of bacteria is coupled. We have also seen the involvement of the sigma factor, which drives the high affinity binding of RNA polymerase onto the promoter. And the sigma factor, second region and the fourth region are important to interact with the -10 and -35 element onto the promoter. We also looked into the details of the RNA polymerase structure and the different channels which are involved. And finally, we have seen the intrinsic terminator as well as extrinsic terminator, and also DNA footprinting, which is normally used to find out the footprint of RNA polymerase onto the DNA molecule. With this, I would like to conclude, and I would like to suggest you some of the reference book which I have used here. So I have used Levin and the Molecular Biology by Nelsons and Cox, and the Molecular Biology by Watson, and also by Weaver, and the Voet and Voet Biochemistry. Thank you for your attention.